MacGregor State High School, and thank you for joining us today as we delve deeper into issues related to COVID-19 and vaccinations. My name is Grace Brown, and I will be your host this morning. We will begin our session with an acknowledgement to country.
Hi everybody, I'm from the Cobble Cobble and Nunokal tribes, and I respectfully acknowledge the Yakura people on whose land that we have this event is taking place.
This morning we are joined by three expert panelists, Dr. Norman Swan, Dr. Larisa Labzin, and Dr. Tom Aechtner.
Dr. Norman Swan attended medical school at the University of Aberdeen and graduated in 1975. He began working at the ABC in 1982 and has played a lead role in its innovation through roles such as the General Manager of Radio National and is the producer and presenter of the Health Report radio program since its inception in 1985. Dr. Swan has also played a key role in TV programs such as Catalyst, Four Corners, Quantum, and The Biggest Loser. He has won 4 Walkley Awards, which are presented for excellence in Australian journalism, with his latest award being for his role in the Coronacast podcast in 2020. Dr. Swan has also been awarded a Media Peace Prize by the United Nations Association of Australia, the Medal of Australian Academy of Science, and the Michael Daly Award.
Dr. Larisa Labzin's interest in immunity started during her honours training at the Institute for Molecular Bioscience. After gaining more experience while working for a research assistant, she moved to Germany to the University of Bonn for her PhD. Dr. Labzin then moved to Cambridge, UK for a postdoctoral training to further investigate how antibodies protect the human body. Her work helps us understand the signaling pathways that are involved in an immune response and what distinguishes a protective inflammatory response from a dangerous one. She is heavily involved in a public outreach and is communicating immunology to a wider public. The importance of this work has been particularly clear during the COVID-19 pandemic, when everyone wants to understand why people are getting so sick with COVID-19. How will we know if we are safe from reinfection when whether vaccines will work?
Dr. Tom Aechtner's research analyzes science skepticism with a focus on vaccine hesitancy, anti-voluntarism, scientism, mass persuasion, and public perceptions of science. He has been a Westpac Research Fellow and a UQ Foundation Research Excellence Award recipient on the project Improving Vaccination Rates in Australia, analysing media, religion, and policy. This has involved researching Australian-specific sources of vaccine hesitancy, including media persuasion and religious concerns, while considering how to positively deliver vaccination information. Dr. Aechtner has developed the world's first massive open online course dedicated to responding to anti-vaccination claims and vaccine hesitancy. He has also developed the UQ Vaccination Choice website, which seeks to provide answers for those looking for information about vaccines. This includes clarity around risks for vaccines, trustworthy information sources, facts, and conspiracies.
So we'll start off with the questions now. My first one is, many of us have heard about COVID-19 started with a bat, but it is hard for us to identify the cause through the different news articles and reporting. Could you please explain the current understanding of how the virus began and how it affected the public trust in science?
Do you want me to start on that one?
Yeah, definitely.
Said Larisa with relief. The reality is we don't know how this virus started. And the World Health Organization recently sent in a team to China to see where it is. And politics has got involved in this because China doesn't want to acknowledge that this, and almost certainly started in China. And that we know pretty much for sure. But they really blocked the efforts of this WHO, this World Health Organization investigative team, and they didn't get some of the key data they needed. I think the smart money, meaning that the best bet on where it's come from, is that and historically, most new diseases in history have come from animals, and they've jumped from animals to humans, whether you're talking about the Black Death, smallpox, measles, tuberculosis. They've all jumped from animals to humans. Even influenza, the flu, jumps from animals to humans. So it's likely that it's come from an animal historically. The one thing that's new is, and bats are they're like a brewing vat for viruses. So bats have hundreds, maybe thousands of viruses inside their bodies. They seem to have an immune system, which Larisa can talk about more than me, which seems to be able to cope with these viruses. And then these viruses exchange genetic information. They exchange DNA or RNA, and new viruses are thrown off all the time. And bats live in colonies close together, so there's no social distancing. They spread it to each other, and you get this mixing up of viruses, and they throw it off. And lots of new infections have come from bats for that reason. So it's likely it came from a bat, and it threw off a virus that was able to infect humans. That probably happens all the time, but this one just happened to be SARS-CoV-2, the name of the COVID-19 virus, and then spread into cities, particularly Wuhan, and there were some super spreading events in Wuhan, which allowed it to spread, and then the rest is history. The other possibility, which is a smaller possibility, but not impossible, is that there's a big research institute looking at viruses in Wuhan in China, and they had been doing, it's complicated, but they're called gain-of-function studies, which means that they actually change the genes in viruses to see how they might change to become much more toxic and contagious. And they had been doing gain-of-function studies in coronavirus, and there is a possibility that it escaped from that lab, but the genetics don't seem to confirm that, but that's still a possibility.
So I can just follow up on some of those points that Dr. Swan has just raised. So when they compare the genetic sequence of the SARS coronavirus to the closest relative is a bat coronavirus. And so that's why they think it originated in bats. And also for the reason that Dr. Swan mentioned, that bats have this really funky immune system, which means that they're really tolerant to a lot of viruses. And it means that they can have all these viruses in them and not get sick, unlike us who get really sick. So what we don't know is when it crossed over. So when did it start first infecting people? And the other possibility is that there could have been lots of times that the virus came from a bat into something else and then into us, but it was kind of a dead end. And then at one point it made a necessary change and mutation, and that meant that it was able to actually infect human cells and replicate in human cells. And that's the kind of like key step that makes it pandemic, because it's then it's able to not just get into humans, but it's able to replicate in humans and pass from human to human. So that's a really key step that SARS coronavirus 2 made, and we're not entirely sure when it made that or how it made that.
That's really cool.
Next question. Do the original COVID-19 vaccines still work on the other mutated strains like the UK strain? And is it possible to create a vaccine that would eliminate all of the strains? And how does the current UQ vaccine work and function, and what is it attempting to do?
Big question.
Yeah.
So quite a lot's known, but not enough is known about how they work with the new variants. So the vaccines work very well with what's called the B117, which is the one that's called the UK variant. It seems to have popped up in Britain. It doesn't work. So the Pfizer and Moderna vaccines from laboratory studies seem to work better with the variants like South African, particularly South African variant. But all the vaccines have shown reduced effectiveness against the South African variant. We don't know enough about the Brazilian one. And the Astra one sounds that it actually doesn't work very well at all, if it works indeed at all, against the South African variant. There's some worrying research into the Astra vaccine. Absolutely possible and unprecedentedly fast to be able to produce new, what are called multivalent vaccines. So that means that you're producing vaccines like the influenza vaccine, which are a mix of different strains, not strains, but mutations of the virus. in one vaccine, and they're working on those now, and some should be available by September. And the UQ vaccine would have worked by injecting a protein directly into the body with an immune stimulant from Melbourne, which would have created a direct immunity to the spike protein.
Yeah. I can also follow up from that if that's all right. So, you know, I like to remind you, all a vaccine is doing is trying to trick your immune system into thinking that it's seen the virus before. And because the structure of the coronavirus is such that it's got this spike protein on the outside, and that's kind of what the virus uses to get inside our human cells. So viruses, they're not like bacteria. They can't replicate unless they're inside, you know, something else's cells. They don't have the machinery or the energy to do that. So if you can stop a virus from getting inside of a cell, you've kind of rendered the virus inert. It can't do anything. So what a lot of the immune response does is really to try and stop the virus from getting inside the cell. And that's through these molecules called antibodies. And so what the vaccines have done is they've basically just taken that spike protein and they're getting it inside the body so that the immune system will make these antibodies and other immune cells that are directed against this spike protein. And they're really good at stopping the virus from getting inside the cells. And so it stops it altogether. The problem, as Dr. Swan mentioned, is that, you know, with these variants, that spike protein has some changes with each of the variants. So that's kind of twofold. Potentially, it makes the spike protein better at binding to our host cells and interacting there. So kind of like making a better key to unlock the door to get inside the cells. But also it means that the antibodies we generate against that original spike protein are no longer as good at binding. And so that's going to be a problem for all of the vaccines because they were all originally made against that first isolate, the one from China. But as Dr. Swan also mentioned, these vaccine technologies are really great, so we can update them really quickly. So either we'll have multivalent ones or we'll just need to get vaccinated more often, like we kind of do at the moment with flu.
With the UQ vaccine, they were trying to introduce the spike protein into the body, but the spike protein is notoriously unstable. So they used a protein to sort of stabilize it and then introduce it into the body. The problem with the UQ vaccine is that the body then created antibodies for the actual protein that was stabilizing the spike. And that's why phase 1 testing, it didn't move past phase 1 testing.
Thank you for your answers. Following that conversation about vaccines, especially COVID, we would like to ask, why are some people against COVID-19 vaccines, and how does this compare with more general vaccine hesitancy?
It's important to recognize that, especially in Australia, approximately only 2% of the population is really opposed to vaccines. And so people often have the misperception that people who are against vaccines, like the COVID vaccines, that population is much larger than it actually is. And that's because people who are against vaccines, they get framed in media quite a bit. It's a much more interesting story to talk about people who are opposed to vaccines than the rest of the population who are sitting on their couch happily accepting vaccines and happily being for vaccines. And this is something called pluralistic ignorance, where people think that there's more opposition to science than there actually is. And that makes people assume that they forget that they're the vast majority of the population accepts vaccines. In terms of the COVID-19 vaccines themselves, we know from past experience that hesitancy around vaccines, so doubt or concerns around vaccines, are always higher with new vaccines and new vaccine technology. So I think that's being reflected in people's COVID-19 vaccine questions. It's just every time we introduce a new vaccine, there just happens to be more questions. In terms of COVID-19 itself, there is a significant amount of misinformation that has been part of the global pandemic from the very beginning. So when we analyze mass media from March 2020 onward, we start to see millions of rumors online about COVID-19, what can apparently cure it, and now a lot of rumors around the vaccines and their side effects. In terms of the reasons why we get low vaccine uptakes overall, there's at least 7 reasons why researchers have found that vaccine uptake can be low. In reality, all the major reasons all go back to distrust. So when people distrust their governments and they distrust vaccine producers and they distrust people like us who are academic researchers, that tends to be the highest the most appropriate indicator or significant indicator of whether people are skeptical of vaccines. So in terms of the COVID-19 vaccine, there's a lot of distrust in governments, and there's a lot of distrust in vaccine producers. And that really relates to people's distrust of vaccines. And that gets fed into by misinformation, rumors about the vaccines, and conspiracy theories around the vaccines as well.
I'm sorry, and the only thing I'll add there is that it's often misunderstood the extent to which there's true anti-vaxxers in Australia. So I think Tom's done a lot of work on this, but the proportion of I think Tom referred to, the proportion of people who are actually anti-vaxxers is very low. Most people will end up getting the vaccine as long as they can be convinced, and the rush to produce them has created a bit of mistrust. And there's a lot of information around. So it's getting the right information out is really important, which is why this session is really good.
So just yesterday, oh, please go ahead, Larisa.
I just had people asking if, they'd be less likely to get the vaccine here in Australia. And I thought perhaps that's different in Australia, where we have managed to contain things pretty well so far compared to if you were, say, in the UK or in the US, where kind of the threat of actually catching COVID is much higher. How do you research that?
So actually, I'm part of a Queensland Health Technical Advisory Group, and we've just ran a study at the end of last year, and the findings have come in this year, specifically in Queensland. And Queensland's also a great case because Queensland has had significantly lower cases than much of the rest of the world. And that has been a concern. So the interesting thing is, I can't give you the data because it's sort of embargoed, but we did find in focus groups and in just talking to people in Queensland that there was sort of a wait and see mentality around the vaccine, because well, we don't really need it because we haven't had any outbreaks. And very few people have actually experienced the repercussions of COVID unless they have relatives. So I had a relative who died of COVID-19 related symptoms in Germany. And then I had a sister who has long COVID in the UK. And when it's personal, people react to it. Now, the interesting thing is, remember we had that snap lockdown. We had focus groups running before the snap lockdown and after the snap lockdown. And people's dislike of masks alone was enough to say, well, I'll get the vaccine if I don't have to wear the mask. And so that in Queensland, that seemed to be the case. But it's true, because Australia has benefited from measures that were effective and have not resulted in the kinds of outbreaks we're seeing, even in my home country of Canada and in the UK where I have family, and in Europe. People don't necessarily see it as an immediate threat. Going back to what Norman said, I just was looking at data from 2 studies that I've been running in terms of survey data, surveying people in Australia. And again, we're seeing about 2% of the population in Australia are ardent vaccine deniers, where we see 98% of the population are kind of quiet acceptors or vocal acceptors. And there's this misperception that anti-vaccination is a much bigger movement than it actually is. And if you accept vaccines, you're actually easily in the social norm and in the social majority.
Well, some vaccines that were in development had to be scrapped because of side effects. Can you explain the process involved in vaccine development, and why the general public should not trust this process?
Sure. Do you want me to start? So I've not been involved in UQ's vaccine development, but I know the team really well. And it's quite a long process. So you start from the beginning, you're testing, first of all, whether you can make the actual bit of the virus that you're trying to mimic. And the different vaccines, actually, all they are basically is different ways of getting that spike protein into the body. So the UQ one, I think, as Tom mentioned, they were just making the protein outside of the body, and then they're injecting that along with something else to stimulate the immune system to recognize that and mount an immune response. With the AstraZeneca one, what they've actually done is encoded the genetic information that tells the body how to make that spike protein inside of another virus, which is called an adenovirus. And so basically that virus which is really safe. They've taken out all the dangerous bits of it. That infects our cells, and it gives that genetic information that encodes the spike into our cells, and our cells make the spike protein. So we turn our bodies into like mini factories that make that viral protein, so our immune system can recognize it. And then the other ones that, of course, got the big interest are the mRNA vaccines. And so they are ones that instead of using a virus to deliver that genetic information, you're actually putting the genetic information as mRNA inside a little fat droplet and injecting that, and that goes into our cells recognize it and make it. So basically all of the different vaccine approaches have pretty much used that as the approach. Some other ones, I think they're mainly in China, they've actually taken the full virus and they've inactivated it. So that's got all the viral proteins, and then they've basically used a chemical so that you're delivering all those virus bits into the body, but it's no longer dangerous. And so all these different technologies have different efficacies. And so the first thing you do is you put them into animals and you see, do they do what we expect them to do, which is make an immune response. So we measure the levels of the antibodies that the animals make in response to that. And then what you do is a challenge study. So you say, okay, they've made some antibodies, they've made some T cells. So theoretically, they should be able to fight off the virus. What if we now expose the animals that have been vaccinated with our candidate to the virus itself? Are they protected? And so that is called the preclinical study, and that has to go first before they can even put it into humans. The next stage is then the phase 1 clinical trial, and that's when they put it into people and see if it's safe, because obviously the last thing we want is for giving someone a vaccine to make them sick. So they do, you know, when we're making a vaccine or a drug, we don't know how much to give to people because we've never tested it before. So that phase 1 study is really where you're testing what kind of dose can we give people that is safe, that they're not going to have any side effects. And also, is it going to make that immune response that we're expecting? Will we be able to measure antibodies and a T cell response in these people who we've given our vaccine to? And that's the point of the phase 1 study. And I think, you know, just from some double checking reading then, there were only a couple fell off. A couple of vaccines didn't make it through, not because they weren't safe, so they didn't cause any disease in people, but they didn't make enough of an immune response. So they compare it to natural infection. They look at people who've had COVID and say, how many antibodies do you make? And they compare it to the people who they were giving their test vaccine to and say, how many antibodies do you make? And as long as the people who are vaccinated make more antibodies and more of an immune response, then that vaccine can proceed to the next stages. And the next stages are the phase 2 and phase 3 clinical trials. So that's when you're giving people the vaccine or a placebo, and then you're essentially seeing how many of those people in a place where there's lots of COVID around, how many of those people get sick. And that's when we started to get ideas about the efficacy. And then, you know, that's when things started to be approved, and now it's being rolled out. It's kind of in what we're calling a phase 4 trial. So, you know, these phase 2 and 3 trials, they enroll something like 30,000 people, because you've got to pick up a big number, firstly, so enough people get exposed to the virus, so that you have enough numbers to know if it's worked or not. And also secondly, because you want to have a big range, people quite different, obviously. So in these phase 4 trials, they're constantly analyzing the data, and they're really refining how effective that vaccine is, how safe it is. Are there small side effects that maybe we didn't pick up in just those small 30,000 people? You know, there might be really rare things that only happen, you know, 1 in 10,000 people, 1 in 100,000 people, 1 in a million. We'll only pick that up in these really large scale trials. So
why aren't all the vaccines 100% effective, and why do some people still contract the virus that they're supposed to be vaccinated against?
So again, great question. And to be honest, we don't really know, but we suspect it's down to everyone's individual genetics. So again, your vaccine response, it really depends on how good your immune response is. Remember, a vaccine is just tricking your immune system into thinking it's seen the virus before. So if your immune system isn't up to scratch, then you're probably not going to have a very good, you know, vaccine response. So there's lots of reasons why your immune system might not be up to scratch. So if you've for instance, if you've got HIV/AIDS, that targets your immune system. So that's one way it goes down. If you've been sick with something else, so quite often people with cancer or with other diseases, they're called immunocompromised. So their immune system is really like run down and they get sick a lot anyway. People with autoimmune diseases is the opposite. Your kind of immune system goes too crazy, and so you might get a bit sick from that. So people with a lot of underlying conditions might not be able to be vaccinated. But for people who are kind of healthy at the start, there's still going to be a percentage of the population who's not going to react to the vaccine. And we think that's down to, your immunity, but it might be a genetic difference. We still don't understand that. So that's important research that needs to happen.
There is an important point, though. It is a great question, but there is an important point here. It doesn't have to be 100%, because there's two things about a vaccine that you've really got to get in your head. One is it's going to protect us as individuals against the disease. So what these COVID-19 vaccines do is that we know that they protect us very effectively against severe disease, getting into hospital or dying. And they protect us less, and a great explanation by Larisa, less effectively at getting any symptom at all, but essentially it turns COVID-19 into the common cold. The second reason you take a vaccine is that you either get no virus, it stops the virus altogether, which is unusual for a vaccine, or it lowers it to very low levels, which means you stop transmitting it. And that's what people start, and then it's not so much what's called herd immunity, where there's lots of immunity in the population. It's mostly that the virus goes down to very, very, very low levels in the community, so that even people who are not immunized get protected by people who are immunized. And that's how childhood vaccination works, is that because 95% of kids in the first year of life get immunized against the common childhood diseases, it means there's very little of those viruses around, even though each of those vaccines is not 100% effective. But because we've got mass immunization, little babies are protected by the fact that we mostly get immunized. There's got to be a little bit of extra cover by family around you getting immunized so that they don't transmit it. But we protect other Queenslanders by being immunized ourselves. So the non-immunized in the community are protected by the immunized.
In terms of vaccine effectiveness, there's also the attribute of some diseases themselves have waning immunity, which means that some diseases, pertussis, whooping cough, you have lower levels of immunity quicker over a period of time. And so therefore the vaccine itself, which has parts of the whooping cough in it, will also have waning immunity. Whereas things like measles, if you get measles, often about 96% of people, if they have natural infection, will have lifetime immunity. And therefore the vaccine itself, because measles triggers a lifetime immune memory in you, the vaccine which has measles in it will also trigger much longer immunity. Some vaccines will induce longer immunity. Live attenuated vaccines tend to produce longer lived immunity than subunit vaccines, because you're giving someone a pathogen, so something that infects you that's in a weakened state rather than just a piece of it. Something called polysaccharide vaccines tend to have, they don't have long-lived memory. And then sometimes it's because their interval between getting a vaccine and getting a booster is too short. And so sometimes there's elements to the actual, what you're trying to vaccinate against. It just doesn't result in long-term immunity or waning, lessening immunity over time just because of the disease itself. And then sometimes it's the type of vaccine. And the reason we give, let's say, subunit vaccines as opposed to live vaccines is because they're safer, and you don't need to have 100% effectivity to bring about community-wide vaccination. So let's give people the right amount of immunity that we can overall in the safest possible package for the overall community.
But the thing about these vaccines is, because of these new technologies, is probably the first time where, and Larisa can correct me if I'm wrong here, but it's the first time where the actual vaccines are better than getting the live infection. So if you get live influenza, not a good idea, you get very sick, you get a much deeper, more profound immunity than you do with the influenza vaccine for all sorts of reasons. But for these ones, the Pfizer and Moderna vaccines, and probably Novavax, I'm just talking about the brand names now and the technologies, are much better than the antibody response you get with a natural infection. Astra, probably not a lot better, but it's good enough to protect against severe disease. So, you could get quite long protection from these vaccines, yet to be seen.
Yeah, it's something that's puzzling but fascinating to us as immunologists, because we want to understand why that is. But I think Dr. Swan just raised a really good point. We're learning about this virus and about these vaccines as it happens. So we're not going to know how long our immunity lasts for until we know. So, it's they last for a year, we'll know in a year's time. How often will we need to get booster shots? We'll know when we know. So yeah, everyone's kind of learning as they go.
The latest studies that came out, the one came out in Nature, I believe, they've found that people that were naturally infected 8 months ago, 95% of the people that were infected naturally still have robust immunity. So the assumption now is that if you are naturally infected with the COV2 SARS, or the virus, you will have ongoing immunity, which bodes well for the vaccines themselves. In terms of vaccines, that other vaccines do seem to produce better results than natural infections. So the HIB vaccine, the HPV vaccine some studies have found that the vaccine produces better immunity than a natural infection. And then also pneumococcal vaccines and tetanus some studies have found that those vaccines actually produce better immune responses than actually getting infected and having the repercussions of being naturally infected.
Yeah.
So following on from that, could the side effects of a vaccine actually be worse than COVID-19 symptoms, and how do we know that?
No, they're not. We know that for sure. Very few side effects from the trials. As Larisa was saying, very large numbers of people in the trials, and now nearly 300 million people worldwide have been immunized. And in countries like Israel and Britain, they've been monitoring the side effects quite closely. So you get sore arms, you might get a fever. If you've had COVID-19 before and you get one of these vaccines, you can get more side effects because your immune response is already topped up, and you may only need one dose, but that's got to be clear. So we know that most of the effects are just minor. With the Pfizer and Moderna vaccines, because of one of the chemicals in it, you can get, if you've got a history of serious allergic reaction, you may get a serious allergic reaction, and but it looks as though there are very few long-term or serious side effects.
Are the COVID-19 vaccines suitable for immunocompromised and people that are pregnant?
Yeah, that's another really good question. So we tend not to do clinical trials on people who are pregnant because they are quite precious, and especially for something that's new, you want to do clinical trials in people that are already healthy. So this is studies that it's really ongoing now. As far as I'm aware, the trials are safe. With immunocompromised people, yeah, that's also a good question. And actually, I don't know the answer to it. I don't know if you know it, Tom or Norman.
I don't personally. No, I don't personally know.
So I think these things are ongoing. It can be, yeah, it's tricky when you're first doing the clinical trials. You really want to make sure that you're enrolling healthy patients, ideally from different age groups and from different ethnicities and backgrounds. But when we start talking about, we call them comorbidities. So that's, other things that people are sick with, and pregnancy, it's not a comorbidity, but it is a more higher risk time. So you don't want to enroll people in something that you're really testing. But now that it's been shown to be safe and effective in healthy people, that's going to be being tested over time. Though I'm not personally aware of any trials that are going on now, but maybe Dr. Swan.
Children, it's been tested in children, people over 12. Pregnancy, there's been quite a lot of pregnant women immunized in the United States, even though it's not approved for that purpose, with no adverse effects. There's increasing evidence that, like influenza, women who are pregnant can get really quite serious COVID and lose the baby as well. So there may well be far more risk in not being immunized in a high prevalence country than being immunized. With immunocompromised people, that's with people with cancer and HIV, you're not giving live virus here. So these vaccines are safe. And it's actually really, really important that these people get immunized, probably with a high performance vaccine like Pfizer or Moderna, so that they get a strong immune response. A couple of reasons for that. One is to protect them against serious COVID-19 disease. And the other is to protect the rest of us from variants. So how variants appear, so it's a bit complicated, but it's related to this immunocompromised situation. Larisa knows 400 times more than me about this, but essentially, if you've not got a strong immune system, HIV, cancer treatment, that sort of, or you're had an heart transplant and you're on suppression drugs, then when you get COVID-19, you don't get a very strong antibody response. And that means a lot of COVID-19 multiplies in your body. It's a bit like a bat. And you get lots, and essentially they change genetic information and they respond to the antibodies, but the antibodies aren't strong to knock it off quickly enough so that they have time to mutate and mutate around the antibodies. So the current theory, and it looks as if this is what's happening, is that the UK variant occurred in somebody who was immunocompromised in England, and it spun off from them. So a variant that was resistant to the antibodies in their body. South Africa has a lot of people with HIV. It's not very well treated. So there's a big immunocompromised community in South Africa, as there is in Brazil. So it may well be that, that you really want to immunize immunocompromised people at a very, very quickly to minimize the chance of new resistant variants spinning off.
Yeah.
I think I'm just going to say that there is actually a bit of a myth that's commonly passed online that states that vaccines are not safe during pregnancy. And this is something that I find in mass media all over the place. And it's actually quite untrue. And so there's actually a special schedule for pregnant women to get vaccinated. And that's because, as Norman mentioned, let's say with influenza, a pregnant woman is 18 times more likely to be hospitalized because of the flu than a non-pregnant woman in society. And so being unvaccinated is significantly more risky than getting vaccines, which have largely not been found to be risky for women. So there's actually a group there's groups of researchers in Australia that their entire intent is to try to get and inform women to get pregnant while they're vaccinated, or get vaccinated while they're pregnant, because they're at such higher risk of certain diseases. And also with things like whooping cough, it's really important that pregnant women get vaccinated to then protect their newborn child. And so it's very key actually that pregnant women get vaccinated. But again, the approvals aren't there that I'm aware of for COVID-19 vaccines.
Thank you. Following from the question that you just answered about if the vaccines are suitable or not, we're curious about the age restrictions on vaccines that have been developed. Why are a lot of the vaccines only for those over 16 years of age?
Again, I think it comes down to what's really been tested so far, and what's been tested in the population. And it's also to do with the risk. So COVID-19 is kind of interesting. Unlike flu, it seems to target children far less. So the younger you are, it looks like the less your chance of developing severe disease with COVID-19, even though you can potentially still catch it. We know that that's not a reason for children to go out and, just party in COVID, because there is still the very small but significant chance that you could get really, really sick with COVID with some downstream symptoms. But I think it really comes down to prioritizing and testing something that's new, that we don't know much about yet, in a population where we know that the immune response is fully developed. But I'm sure that this will be tested later on, because the idea will be to get people vaccinated as soon as possible.
Dr. Swan, Dr. Labzin, Labzin and Dr. Aechtner. Thank you for your time this morning, helping us understand COVID-19, vaccines, and your human hesitancy to the vaccines. I think that I personally found it really interesting, and I think it's helped a lot with some of the questions that I personally had. We look forward to learning about this further and sharing our insights with others. Thank you.
Thank you very much. Good fun.
Thank you very much.
Thank you.
Thanks.
Bye.
Bye.
Bye.